of Alzheimer's disease in particular is that we didn't really know what caused it. And a lot of the research that I and many of us in the room has done has been trying to identify the causes so that we can intervene and improve the outcomes. So if you've got any questions during the talk, please do feel free to put your hand up and ask questions. I don't have many conflicts, but there they are. So one of the most potentially infuriating things as an epidemiologist is that we have a lot of data about the risk of disease in the population, the consequences of different diseases. And the reaction of the population, the reaction of governments to different conditions that have very similar end results are wildly different. So just in preparing for the talk, I thought it'd be interesting to compare the response to COVID to the response to the dementia pandemic. COVID around the world caused about 7 million deaths. Dementia causes about 1.6 million deaths per year. So every 4 and a bit years, we have what is equivalent to a COVID pandemic solely from dementia. And we all know the difference in response in terms of funding. In the UK, we spend £15 billion a year on care, primary and secondary care for dementia. We spent somewhere in the region of 300 to 400 billion on our response to COVID. These are just completely different orders of magnitude. We've had risk factors for dementia mentioned earlier today, and I'm putting up this example from the Lancet Commission, who have published a series of reports into the risk factors associated with Alzheimer's. And the evidence that they used for basing this report is largely observational epidemiology. So they have investigated the associations and brought together all of the evidence, the best evidence that we've got from observational epidemiology about the risk factors that associate with dementia. And I should say at the beginning, I'm particularly focused on Alzheimer's disease rather than vascular dementia, for reasons that will become sort of clear. Of these risk factors, they list the ones that are shown on the slide, and they quantify the population attributable risk. What is this? This is the proportion of cases in the population that can be explained by any given risk factor. And the argument goes that if we change these risk factors, we can reduce the burden of disease in the population. So you can see that we've got education attainment is one of the largest established factors, but there's a large number that together explain about 40%. The question is whether these risk factors that associate with Alzheimer's disease are causal, and whether modifying them is going to have any or is going to result in this size of effect that we see here. What's missing and often not included in these reports is genetics. We've had it mentioned a few times today, but the ApoE variant has been identified many years ago as the single largest risk factor for Alzheimer's disease. So relative to the evidence that we just saw from the Lancet Commission, where the observational risk factors can explain around 40% of the variation in Alzheimer's disease, what is the population attributable fraction that we can attribute to the ApoE variant? I should say that this is work, very recent work that Dylan Williams, who's sat over there, has been doing jointly with Emma Amundsen as well. So we're kind of curious what, I guess, what response we're going to get to this one. Um, we looked up the ApoE variant in the UK Biobank, where we've got a sample of individuals with linked health record data. We have diagnoses on Alzheimer's disease, and we've now got between 12 and 16 years of follow-up up to December 22. So we've got a fair bit of follow-up, and we've got clinically ascertained cases. These cases won't be perfectly ascertained, but they are likely to have Alzheimer's pathology. The other thing to note is the frequency of the different variants. The number of individuals who carry any of the ApoE risk variants is remarkably high. This is important for the population attributable risk. So 95% of people are going to have one E3 variant, and almost 30% of people are going to have up to one E4 variant. Individuals with the highest risk, those who are homozygous for E4, these represent about 2.4% of the population. So there's a large number of people with these variants. Previous estimates of the population attributable risk for ApoE have focused on the risk attributable to E4 relative to E3. But really, that's not the question here. The question is relative to the lowest risk variant, which of course is E2. So first of all, what's the increased risk in Alzheimer's that we see from the ApoE variant? Well, I think this is very, very well known from the literature. It's huge. So having one additional E3 or E4 variant increases your risk of all-cause dementia by about 74%. Being homozygous for E4 relative to being homozygous for E2 increases your risk of all-cause dementia by 10-fold. These are enormous effects by genetic standards or even phenotypic epidemiological standards. And these are estimated with a huge degree of precision, given the sample size that we've got from the UK Biobank. So what about for Alzheimer's specifically? Here, the effects are even larger. We've got a odds ratio of 4, so 400% increase in the odds for having a E3 or E4 relative to E2. And you have a 25-fold increase in the odds when you are homozygous E4 relative to homozygous E2. So these are absolutely enormous differences in the risk of disease. So what does this mean for our population attributable risk? Well, we have the formula for this, and we can estimate the population attributable risk. For Alzheimer's disease, we estimate that 73% or almost 74% of the population risk of Alzheimer's can be attributed to the ApoE variant. Similarly, for all-cause dementia, it's naturally lower because this is going to include a greater degree of vascular pathology, but it's 40%. So a very, very large fraction of the total burden of disease in the population can be explained by variants in the ApoE gene. We compare this to the observational results that we get from the known risk factors. These are far, far larger. So the first point that I want to make, and possibly the most controversial, is that should we rename Alzheimer's disease to ApoE disease? On one level, this is super easy, right? Because we only have to change one word in our paper and it would all just follow through as AD. But if the pathology and the origins of the risk of the disease comes largely from a single variant, that should be the focus of the causal inference that we're doing and the attempts to cure the disease in the population. So if we're going to mitigate and reduce the risk of Alzheimer's disease, we have to reduce the risk and mitigate the effects of E4 and E3. Okay, so that's where the risk in the population is coming from. It's coming from these genetic variants, or a very large fraction of the risk of Alzheimer's is due to these variants in this gene. The next question is, well, when does it have its effects? We've had a question earlier. I think a number of people raised it, memorably by Paul, when he was trying to get funding for studies of younger individuals at risk of Alzheimer's disease. So the question is, when should we be treating people who are at increased risk of Alzheimer's disease? And when do the effects of these variants appear? One way we can do this is in large studies like the UK Biobank, where we've got individuals who have been sampled and phenotyped across a range of ages. We know from the transmission of genetic material from parents to offspring that the distribution of genetic variants within families, at least, is going to be largely random to with respect to the environment that they experience. And this is the principle that underlies Mendelian randomization, and we can use it to learn about the causal effects of genetic variants and risk factors in the population. Now, traditionally, Mendelian randomization studies work from exposure to outcome. We attempt to investigate a risk factor for disease, and we want to know if it affects Alzheimer's. These have been very widely used as an approach to investigate risk factors that might causally affect Alzheimer's disease. Today I'm going to talk to you about a study that was conducted by Roxana, who's my former PhD student, who's in the room, that was recently published in Nature Communications, where we wanted to reverse this approach. We investigated the links between genetic variants associated and the polygenic risk score for Alzheimer's disease and the phenome. So we conducted an outcome-wide phenome-wide association study. And what we were particularly interested in is trying to identify when phenotypic differences between individuals at high risk of disease and low risk of disease occurred. And to answer Paul's question, we looked through all the different phenotypes that were measured in UK Biobank, and then estimated the association between polygenic scores for Alzheimer's disease and each of these phenotypes. And the hypothesis is that the earliest manifestations of the disease process are commonly indicated by the variants that were indicated as associated or found to be associated with this polygenic score as early in life as possible. And hopefully the results are interesting. One thing to note about the UK, well, there are many things to note about the UK Biobank study, which is remarkable for lots of ways, lots of reasons. First of all, the selection into the study is incredibly strong. So here I'm going to be reporting on results which is restricted to the European subsample. It represents over 300,000 people, which is an enormous sample, but is a very small fraction of the individuals who were originally invited to participate. We know that these individuals are population, and if there's time, I'll come on to that at the end. So this study had two broad steps. The first step was to investigate the relationship between the Alzheimer's polygenic risk score and the phenome in these different age tertiles. Then the second step, we followed up each of the phenotypes that were detected in the first stage in a bidirectional Mendelian randomization study. So we investigated whether any of the phenotypes that we detected were causal. And the intuition is that we have, in the second stage, was that we have genetic variants for each of the risk factors. SNPs have been identified in external studies where we're able to instrument for the exposure that was detected in the PHEWAS, and then subject to the rule, the assumptions underlying Mendelian randomization, the three most crucial being that these genetic variants associate with the trait, which we can test and prove. Second, that there are no confounders of the association between the SNP and Alzheimer's disease. And third, that there's no direct effect of the genetic variants on Alzheimer's disease. We can test this hypothesis to generate an estimate of the effects of each of the phenotypes on risk of disease. Now, the outcomes were very interesting in that for the physical health measures, here we have the youngest samples in black. We have the middle-aged individuals between the ages of 53 and 62 in blue, and then we have the oldest individuals in red here. And for multiple phenotypes, we see differences as individuals age, as this relationship between the Alzheimer's score and the phenotype increases. And across each of the physical health measures, we see this relationship. Because we're doing this phenome-wide, we're doing it systematically across all of the phenome that's measured in UK Biobank and was available in our software package. And we see consistent changes where higher values of the Alzheimer's score were associated with lower values for each of these physical traits. So lower diastolic blood pressure index. And these associations increase with age. We see differences in cognitive measures incredibly early in life. So if we're interested in when Alzheimer's pathology, or at least ApoE pathology, emerges, it's likely to be in the youngest ages, already present and already developing, which only increases with time. So they're slower to complete some of the cognitive testing, and these deficits increase as the sample ages. We're seeing differences in numerous biological measures very, very early in life, and these differences are large and consistently detected. We also replicated these in the Hunt study. So what did we see when we looked in the reverse direction for the phenotypes that we picked up? We found evidence for effects of the Alzheimer's PRS on a significant number of traits, but for the vast majority of phenotypes that we investigated that aren't indicated on this table, we found evidence that they influence risk of Alzheimer's disease. So if we kind of go back to our Lancet Commission evidence of the risk factors for dementia, for Alzheimer's at least, we can kind of get a test from Mendelian randomization of which of these are likely to be causal. So we had evidence of hearing loss was listed as one of them. We have hearing aid user in the UK Biobank, no evidence that it associates with Alzheimer's disease. Systolic blood pressure. We have a confidence interval from 0.99 to 1 with a p-value of 0.49. Alcohol investigating the UK Biobank because we didn't have sufficient measures or variants to instrument it, but Andrews in 2020 did investigate this more using different phenotypes and different samples and found very little evidence of a causal effect. Obesity, BMI, very little evidence of an effect, relatively tight confidence intervals. Smoking, results much less precise, again, no evidence of an effect. Depression, we weren't able to look into it, but Hung et al. 2020 did, and found very little evidence of major depression, an effect of major depression on Alzheimer's disease. Social isolation, we had relatively, these social variables are harder to use with Mendelian randomization, but again, they were less precise, but didn't find any evidence of an effect. Physical activity, moderate physical activity was actually implicated in a higher risk of Alzheimer's disease, and there was very little evidence, albeit imprecise, for vigorous physical activity. Diabetes, we weren't able to investigate this, but other studies have, and again, at best, a modest evidence of an effect. So we're looking from genetics and Mendelian randomization. The only variable that we were able to find compelling evidence of a causal effect on Alzheimer's disease was educational attainment. So my conclusion, at least from the genetics and from the evidence from Mendelian randomization, is that the vast majority of these risk factors are probably not causal. So where does that leave us? Just because something is genetic in origin, or we're able to explain a very large fraction of the variation in the population attributable risk, doesn't mean that we can't intervene on it. There are a very large number of phenotypes and clinical outcomes which do have a very significant genetic basis. And I wanted to take inspiration from the experience of cardiovascular medicine over the last 50 years, which in Bert's talk earlier, that there was a huge reduction in the rate of cardiovascular disease, some of which was explained, some of which wasn't particularly well understood. This quite nicely mirrors the situation for Alzheimer's disease and dementia that we've heard mentioned earlier today, that actually the development of treatments for cardiovascular disease, particularly focused on the discovery of statins, which occurred in 1976 and was commercialized and introduced in the late 80s, actually occurred after a lot of the reduction has occurred. As we've heard earlier today, the rates of Alzheimer's disease and dementia in the population, or the incidence of Alzheimer's disease in the population, has already started falling. So, in summary, we know what causes Alzheimer's disease. Largely, it's genetic variants in the ApoE region. We know when its effects start to be phenotypically expressed, appears to be midlife. So this is not a disease of old age. This is a disease that's pathology is detectable in individuals who are far, far younger. So we are now in a situation where we have the first treatments that show any evidence of efficacy on any of Alzheimer's phenotypes or measures. However, a lot of the treatments it's not clear if they're going to be effective, and it's not clear whether the side effect profile of them is going to be acceptable. So we're in a position where that's likely to change over the next 5 to 10 years, where the effectiveness of these treatments is likely to be refined, as was the case with statins 50 years ago. But the things that we need to optimize as epidemiologists and public health physicians is who are we going to treat, how are we going to treat them, and how much do we want to spend, and how much resource do we want to dedicate to resolving this problem. This seems to be something that we could solve as epidemiologists, and it could be something that we could solve relatively soon. So if we are thinking about a program to eliminate Alzheimer's disease from the population, the people who we want to treat, at least in the first instance, are going to be carriers of E4. Homozygous individuals would be the primary, would benefit the most. We probably know when we want to treat them, but it's probably a good idea to start a hell of a lot earlier than many of the studies are doing at the moment. But in order to do all of that, we need evidence, we need health economic studies to justify, I guess, the spending and the clinical interventions. Kind of given a brief overview of those two studies, there's a lot of limitations to this work, principally representativeness, measurement error, and case ascertainment. Statistical power of the Mendelian randomization studies were underpowered. With better powered studies, we might be able to detect effects on some of these risk factors. And for some of the risk factors, some of the potentially most important risk factors, potentially infections, potentially air pollution, we simply can't use a program like Mendelian randomization. Thank you very much for listening. I'm curious about what you all make of it and what you think.
I was going to ask, to what degree do results from MR correspond to twin studies? For example, I've seen studies in monozygotic twins where depression is linked to increased dementia risk. If results from twin studies and MR contradict, which evidence would we say is stronger?
That's a great question. So it depends on the specific research question. Twin studies are often, they're harder to use for research into dementia and Alzheimer's simply because of the age of the cohorts. So twin studies tend to be a lot younger. For the specifics of your question with the link between depression and Alzheimer's, I don't know. And the theoretical, which would you prefer? I think there are reasons to prefer. Again, I'm going to be controversial here, but I think that the evidence that we get from Mendelian randomization with genetic variants, that we know that there is a source, an exogenous source of variation, that randomness that occurs at conception is very unusual in clinical medicine, and is a very powerful source of evidence about the causal links between these different factors. It does have limitations, particularly when you are interested in phenotypes and clinical outcomes like depression, which is a very heterogeneous psychiatric condition. It's one of the hardest hypotheses to address with Mendelian randomization. And I guess ideally we do want to be triangulating and extracting evidence from all of the different approaches rather than putting all of our eggs in one basket.
Thanks, excellent work. I really like your approach. But you're a statistician. Can you say something about this kind of lack of power? Because you're sort of putting yourself in a position to prove the null hypothesis. You know, and how likely are to find using genetics to find like smoking? You know, how much of the variation is smoking? Can you explain by genetics? And then if you don't find anything here, how can we then say that the Lancet Commission is wrong? Where do you think the Mendelian randomization can have the best use case?
Okay, another excellent question. So I say I've argued that the Lancet Commission is wrong, and I'm using the Lancet Commission as example in the field, right? It's an incredible piece of work where they've got comprehensively summaried a huge amount of data, observational data, but we know there are flaws with that approach. As epidemiologists, we know that observational studies can suffer from residual confounding. We know that they can be very unreliable guides to causation. Further, we know that a lot of the trials which have been designed on the basis of observational data have not been effective. So to date, we don't have randomized trials for a lot of the phenotypes that have been measured in observational studies that associate with Alzheimer's disease in observational studies. The randomized trials that have been designed on the basis of that, be they diet, be they exercise, there isn't one that's shown a huge effect on Alzheimer's. So I think there are real questions about those epidemiological approaches that as epidemiologists, we know the limitations of this research. And so taking and drawing inferences from those sorts of studies to try and learn about the causes of disease in the population is going to be challenging. And I think that's where the appeal of other approaches, not just Mendelian randomization, but other approaches that take the identification of causal effects as seriously as possibly and as the core goal. So to try and cause of naturally occurring events, which can give us information about the causes of disease in the population, is really valuable. Finally, just to get onto your point about power, like you're absolutely right. For a lot of these, the power is not enough to exclude a clinically important effect, including the size of effects that's being reported by the Lancet Commission on observational studies. So I think so some of these are going to be overlapping with the observational estimates, and you can see that in the precision of some of these estimates. So if we've got an odds ratio of somewhere between 0.6 and 1.22, that includes a pretty big effect. The other thing to say about smoking, of course, is attrition bias, and it's going to be a real issue here, right? Because if you're a smoker, there's every possibility that you're not going to live for long enough to develop Alzheimer's disease, and so you're going to have selection out of your study, and Mendelian randomization is going to do absolutely nothing to protect you against that. In fact, could make it worse. But for some of the other exposures, particularly systolic blood pressure, this is not a lack of evidence here, right? If we've got an odds ratio that is as precise as that, similarly for BMI is getting pretty close to that level of precision, that these are not null findings. There's no information here. These are null findings. They're precise enough to draw a conclusion and say this is probably not a credible cause.
Yeah, okay. I have to react briefly to the Lancet Commission because I use that in my teachings as an example of very bad research because, and I say, typically I will say what they are right about is that there is a lot of non-genetic influences that may accumulate over the life course, and otherwise it's just associations that they put together. And I think maybe it is because there was a lack of real epidemiologists in the Lancet Commission, but that was not my question. I was actually going to ask you about ApoE because I do agree that that is of course an important risk factor, but I was a bit surprised by that you said that it's the cause in 75% approximately of all cases, because at least in some of the older studies showed that the majority of Alzheimer cases are E3 carriers. So how can you explain that? And maybe that has also to do with this concept of population attributable risk, where people typically, well, you have a percentage, but I mean, the total is not 100, right? The total may be thousands or going up depending on how much we do. So I would really like to hear your comments on this.
Yeah, another great question. So your first point about the E3s, E3 is not a neutral allele when it comes to Alzheimer's risk. E3 is a risk allele. It's just better than E4.
question was, you say 75 to E4. And if 75% of all AD is due to E4, then I would expect that at least 75% of all AD cases have E4, because it's very hard to be due to something that you don't have.
In which case I explained it wrong, and I apologize, because that is not what I was intending to express. So what have I written? So this is a population attributable risk to ApoE, E3 and E4 relative to E2. So it's not the population attributable risk to E4. It's the attributable risk to both of those variants versus being E2. And if you look in the...
So E2 is not taken as the reference, but as one of the risk alleles.
And I think it was even another slide where you mentioned that it was 75%.
So E2 is our reference. That's our lowest risk of Alzheimer's disease.
One or two copies of E3 and E4. So 99.4% of the population, the one or two copies of E3 or E4, they're on average conferring about 4 times the risk of AD, at least, you know, with the consensus in EKB data. So almost all the population have a moderate to higher risk of Alzheimer's due to E3 or E4. That together equates to about 74% of the burden of AD. Well, it is the population attributable fraction if you were to obviate the effects of E3 and E4 coverage. Which, sorry?
Gray hair causes Alzheimer's.
Well, what do you mean by grey hair?
90%.
Yeah, sure, yeah.
Grey hair.
Well, yeah. I mean, obviously, we need a lifetime risk to develop AD, but I mean, it doesn't preclude there being a lifetime cause of that. So.
Biology and compare E4.
That's the exception.
Because E3 is what most people
I like this discussion, and this is where causal inference and causal inference thinking really come into play, because I think the very first point we need to make with population attributable risks is that it actually only makes sense to talk about population attributable risk when you know you're studying a factor that is intervenable. ApoE, it's not. So basically, this whole discussion stops there. But let's, for the sake of hypothetical argument, go further and say what it means. We really need to be careful not to compare population attributable risks calculated for a single risk factor, such as ApoE, and compare that directly to a population attributable risk that is calculated based on a combination of risk factors. Because that single factor population attributable risk, you know it's going to be inflated, because the population attributable risk does not take into account interaction, what Bert Hofmann so eloquently explained this morning, that one disease case could be due to 2 factors. That's not being taken into account. Consider, for instance, a stroke. You could suffer a stroke because you both have an embolus atrial fibrillation as well as small vessel disease, because only then the thromboembolism gets stuck there. And because you're alive, and because there's oxygen, because all these factors have attributable risk of 100%. So calculating population attributable risks of a factor separately, you can do that, but don't make that interpretation of, hey, if I would prevent this, I could prevent this many cases, and definitely not compare it to a combination. We actually did this exercise in Rotterdam study in which we more or less took the same factors as a Lancet commission. We came up with a population attributable risk of about, I think, 34%. And if in that combination we would add ApoE, it only increased that population attributable risk by 6%. So then the argument becomes, oh, ApoE, it can only be can only prevent 6%, given there is interaction with all those other factors. So I would, I like the rest of the presentation, which was very provocative about the Mendelian randomization. I kind of am criticizing this start with the population attributable risk.
Yes.
If there's time. I have two things. One is, of course, I agree about using Mendelian randomization to exclude causality. I have, especially if it accounts for 5 or 6% of the observed variability. I have concerns with that. The other two extensions you made about these drugs are going to work in midlife and in ApoE, that's absolutely dangerous because, as you know, having an E4 increases your risk of side effects. About 33% of people on E4 are going to have brain edema, potentially long-term gliosis if squirrel monkeys are anything to go by. And only now we've started having some studies in preclinical asymptomatic people. And we have no idea if it's going to help them or hurt them. And this is, we are talking about 65-year-old preclinical people. So, and the total benefit from these so-called wonderful drugs is about 4 to 7 months slowing down in the slope. So, which is exactly the same as what donepezil does. It's symptomatic. This is supposedly disease modifying. Over time, now there are some emerging results, and I hope to God these are miracle drugs, but they are not penicillin for pneumonia. And I'm very concerned about sort of saying midlife for interventions that have such high toxicity. Let's not.
So, yes, and that's where I think the example of statins is quite apt and quite beautiful here. So if you look at statins, they were developed in 1976 or discovered in 1976. The first statin to be discovered, you know what it was? It was discovered in Japan, and it was demonstrated to reduce LDL cholesterol. Unfortunately, it had side effects. And in the mid-80s, the concerns about the effects of and potential side effects in animals. The first molecule that was developed was not effective. The side effects were too severe, and so it couldn't be used in humans. But as a strategy for treating disease, primary intervention in midlife to prevent cardiovascular disease in later life is absolutely where we've got to. We've only got there through billions and billions of dollars of research funding. So we 100% have to do this properly, and you've got to go through that scientific process. But as a strategy for treating disease, particularly primary prevention, given what we know about the pathology of Alzheimer's disease and its phenotypic expression in much, much younger people, it seems certainly a fruitful avenue for future research. But I guess we'll see over the next 20 years, right?